Yeah. Hello, everyone. Thanks to the organizers for giving me the opportunity to speak. I'm Michael. I'm a postdoc in the lab of Jonas Friesen at KI in Stockholm, Sweden. And we are interested in... So our motivation comes from our interest in stem cells, and particularly we study a process called adult neurogenesis, where we very often use a tool which is based on in vivo clonal tracking to identify the progeny of all progeny of a single cell. And while this is a very powerful tool, it really suffers from low throughput because we rely on sparse labeling, and it doesn't really provide any molecular information apart from a few markers that we can stain for with antibodies. But on the other hand, of course, we have technology like single-cell RNA sequencing, which is enabling us to profile thousands, even millions of cells in one assay, and basically at high throughput obtain whole transcriptome information. But often we don't have lineage information, and very often we also rely on dissociated cells. So as other speakers already introduced before, we're basically we want to solve that and basically combine the best of both worlds by introducing expressed genetic barcodes into brain cell progenitors that then can be captured later on using RNA sequencing. And I think these cellular barcoding approaches in neuroscience there can be very powerful, and I think there are like three very big areas where this can contribute to. I think what most people study is, of course, how diverse cell types are generated from a very homogeneous pool of progenitor cells. I think what's really interesting too is to relate gene expression to neuronal connectivity and ultimately activity and behavior. And I think what's also really interesting is to study how neuronal lineage and birth timing influence ultimately wiring specificity. And I think lineage tracing or clonal tracking coupled to RNA-seq is very powerful in that area because we don't really have a good understanding of the molecular mechanisms underlying this. So our contribution to this area is an approach that we call TRACS, and this stands for clonal tracing and expression profiling in the mouse brain using single-cell RNA-seq, and relies on a lentivirus library similar to what Klein described or SMF Morris also was working on. It's basically a lentivirus that encodes a nuclear localized EGFP. In the UTR, you have a barcode, which we call a clone ID, and this is upstream of a polyA site for single-cell RNA-seq capture. And the way we barcode the cells is we use a ultrasound-guided in utero injection. So we're interested in the developing mouse brain. So we inject roughly 1 million barcodes into the mouse forebrain at embryonic time point E9.5. That's just before neurogenesis begins. And we think that under our experimental conditions, we have most of the cells uniquely labeled with only single clone ID. Following our injection, we wait about 3 weeks and then isolate cells from the 2-week-old mouse brain, where we then isolate barcoded cells using FACS from 3 different areas. And we use droplet-based single-cell RNA-seq to reveal the transcriptomes and also the barcodes. And here's the entire data set, which is corresponding to 4 brains and a single control. We have around 62,000 cells, many diverse cell types. So there's 40 different cell types coming from these different regions, which is the cortex, the striatum, and hippocampus. So these are 4 brain regions of the mouse brain. And we see that there are some cells which are, of course, found in all 3 regions, but many cells are also region-specific. For example, glutamatergic excitatory neurons we mostly find in the cortex, or radially-like stem cells, mostly in the hippocampus. And because we're the non-injected controls, we first assessed cell health just to see whether lentiviral barcoding somehow messes with the cell physiology. And we first correlate average gene expression between classes of cells of barcoded and non-injected controls. We see there's a very high correlation. And we also basically investigate whether we have certain immune response genes that are associated with an HIV infection, which the lentiviral barcode is based on, or the lentivirus genome. And we can see an obvious upregulation of such an immune response genes. Genes that we see whether differentially expressed between barcoded and non-injected cells are, for example, EGFP, which of course makes total sense because it's a transgene that we introduce. So from that, we subset our data on only our barcoded cells, which are roughly 50,000. We extract clone IDs from the data, get from something like 19,000 cells, we get clone ID information. Most of the cells have like a single clone ID, something like 10% contain 2 or more. And then we can now use these data to reconstruct clones. And overall, it's about, it's more than 2,000 clones and 12,000 cells in these clones. And I just want to point out here that if you would want to collect data of 2,000 clones using classical clone tracing, you would definitely end up with sacrificing hundreds of mice because you really rely on sparse labeling. We find that 25% of our total population of cells are containing clones. Since we have so many different subtypes of cells, this of course varies across cell types. But I think what's one interesting aspect is to look at the general clone size. And we saw here like a dramatic difference between cell types that originate from, that are mesoderm derived, like D cells. These are microglia and perivascular macrophages. And they have like roughly 8 times bigger, they make 8 times bigger clones than neuroectoderm derived cells, such as neurons or glia cells. And we think this makes sense because there's only a small window where these cells basically migrate into the brain. So you have a very small population of progenitors that then probably needs to increase the clone size rapidly to then have enough cells to populate the entire brain. Another aspect we looked at is the regional distribution of these barcodes, these clone IDs, and we find that most of them, they accumulate in one of the 3 regions. And if you find clones that are regionally dispersed, this dispersion often happens between the region of the cortex and the striatum or cortex and hippocampus. And now we zoom in a little bit into the cell types that are associated with these more diverse or dispersed clones. And that's basically quantified here, where we have the percentage of cells that are spread across more than one region. I just would like to highlight 2 examples here. So one region that is barely crossed is between the striatum and the hippocampus, and we find clones that do that, and they are most of the time associated with oligodendrocytes. So these seem to be very migratory cells that we know are generated in this part, in the ventral area of the brain, and then they seem to kind of migrate out and populate other parts of the mouse forebrain. And what we also came across again were, again, microglia. These seem to be the only set types that are very migratory. So they don't only produce huge clones, they also are very migratory cells that seem to enter the brain in one location, and then from there kind of spread out over the entire tissue. Another aspect that we were interested to look at in the data is the fate distribution of clone IDs. And here we use a tool that was also introduced by Klein in the science paper in 2018. And we calculate what we call the clonal coupling score, which is simply the number of shared clone IDs relative to randomized data. And when you do that, you see that you have in that heat map, you basically get 4 groups or 4 clusters of cells, and they mostly cluster by region. And I would like to draw attention to this hippocampus cluster, which itself is containing 2 smaller subclusters. And here we think we are talking about 2 different progenitors, fates, and fate 1 is a progenitor cell that mainly makes astrocytes and neurons, while there's a second fate that is already biased at a very early stage, which is E9.5, just before neurogenesis begins, that just makes astropendular cells and oligodendrocytes. And finally, as I told you, we would like to correlate the transcriptome also to the connectome, or even read out neuronal connectivity using transcriptomics. And for this, we use, of course, in situ sequencing, and particularly we use spatial transcriptomics. And our approach that we call space tracks, because it's transcription expression profiling in situ on space, allows us to read out clonality, cell type, and transcriptome. So we take a barcoded brain, make tissue sections, we mount them on a glass slide, fix them, and do an immunostaining step, which allows us to essentially reveal the cell type, the broad cell type of barcoded cells. And this is how the data looks like. We, as I said, from immunostaining, we can distinguish between major cell types. In our case, it's neuron oligodendrocytes. We can reconstruct clones, and interestingly, they're significantly larger than when you use the TRACS approach, which relies on dissociated cells. And that's probably because we, of course, don't dissociate the cells, and this leads to a reduced cell loss. Overall, we see a similar pattern that we have an accumulation of clonidines in major brain regions, but now we can also zoom in and see that there's like large dispersions within these regions and information that we couldn't assess before. And finally, we can also reconstruct kind of classical clonal migration patterns that were described previously in the mouse brain using this technology. So here you have dispersed clones, they're far spread over the tissue, and you have also these clusters like radially oriented clones in the cortex. And as an outlook, I would just briefly mention that we can actually also study this, use this technology to study the clonal maintenance of gene expression. So we ask how clonally related cells, or how similar they are, and whether they maybe share heritable gene expression states. So we basically look at gene expression similarities beyond cell types. And I'm pretty excited about this data because we see, for example, that one of these clonally expressed genes is the transgene that we actually put into our cells. So I consider this a positive control because each clone shown here in the violin plot basically is represented by one progenitor cell. So it makes sense that between progenitors we have slightly different clone and EGFP expression levels because the transgene is integrated into different loci. And another gene that we find in these radioglial-like stem cells in the hippocampus is, for example, claudin, which is a component of tight junctions. And a final example is a molecule called the cell-adhesion molecule 2, which we see in, say, 1 glutamatergic neurons in the hippocampus, and which seems to be kind of clonally maintained between related cells. So in summary, I showed you a technology that we use for retrospective clonal tracking and expression profiling in the mouse brain. After a one-time in vivo barcoding step, we identified unique features of brain macrophages such that they make these ultra-large clones that are regionally dispersed, and these cells basically have a single progenitor origin. We see already very early, just before neurogenesis, 2 populations of fate-biased progenitors at the hippocampus. And we introduced a technique called space tracks to simultaneously do clonal tracking and expression profiling in situ. And we also try to identify first kind of genes that are associated with clonal gene expression. And this brings me to the end of my talk. These are the people that contributed to our work. That's our lab in Stockholm. And thanks for your attention.
Thank you, Michael. We have a question from Maria Luisa Sequeira Lopez. It says, fascinating approaches and data. I agree. Do you see any developmental or iatrogenic effects in postnatal life, including seizures due to direct injection in the embryonic brain?
Yeah, I can't say. I wouldn't expect it because I think it's because we don't track the animals until they're actually adults. So we usually sacrifice them after 2 or 3 weeks after birth. But I wouldn't suspect it because I think these neutron injections are very widely used. Other people use it for much longer time points, and I wouldn't expect any detrimental effects from that point of view.
Maybe I'll ask a question as well. It's also sort of a technical one. I'm curious, to what extent does the accessibility of cells to the injected material bias what cells are available for you to be able to track using this method?
Yeah, that's a good question. So right now we do this intraventricular injections. So basically into the lumen of the ventricle, which is volume in the developing brain, and we mainly target cells that are, of course, facing this ventricular volume and the ventricular zone. For us, that's great because these are the actual stem cells that we're interested in. I think at later time points, it becomes a bit more difficult because this volume becomes very tight. But what we're also doing, we're working, for example, with viruses like AAVs that spread more within the tissue, and it can actually also be coupled with transgenic lines to target other cell populations.
I think we have to move on to our last speaker. I encourage people to look in the forum to answer any other remaining questions. Our last speaker of the session and the conference, I guess, is Merit Romaika from Max Labs in Vienna.